Welcome to Snap Podcast.
Welcome to another episode of the Snap Podcast. Today we have Metka Lenassi on as a guest, and we're going to be talking a little bit about her history in EV research, her time spent on the ISEV board, and a bit about the recent EV Corona workshop that she helped with organising. So thank you for agreeing to speak to us, Metka.
Hi, Liam. So it's my pleasure to be here, and I'm just amazed about SNAF and all of the activities that the Student Network for EV does. So really, congratulations on everything that you do, and glad to be here.
So do you just want to start off with telling us a little bit about how you got into EV research in the first place?
So that was an interesting story, actually. So my PhD topic was on fungi. So I was studying stress signaling in fungi. And for my postdoc, through my mentor, she organized that I could get a place at UCSF in Metka Lenassi lab, who was working on HIV, and they had a lot of different projects on regulation of transcription and other factors. But then there was this one small project on EVs. This was in 2007, so it was in a way early in the EV field. But the person who was doing that project for a year before me actually quit and didn't want to continue on that. So everybody was warning me like, "Oh, this is a dead end project. Don't do that. You'll just waste your time." But when I read about this, it was cell biology, biochemistry, all the stuff that I love. So I was kind of like, you know what? I'm going to do it. I'm going to try hard and let's see where it goes. And in the end, it was actually, at least I was very proud on this publication. It was we were the first to show that NEF, HIV virus, has a accessory protein NEF, and this NEF induced release of extracellular vesicles from T cells, and these vesicles that contained NEF caused apoptosis in bystander cells. So this was very early on, and it was published in Traffic, and it was just something you're very proud of, especially because the start was very it was a risk. But I mean, in the end, it became my career. And actually, when there was this, I was applying for some projects, and there was this one reviewer who actually commented that you have a good insight because you knew which field will maybe be interesting in the future, and you went there and changed the career, and just you had a good instinct. So I was okay, maybe it was fun.
Was there much of a Slovenian EV community at all at the time?
I mean, at that time, actually, there was at least, it's Slovenia is 2 million people, so of course there is already a limited number of scientists.
Yep.
But I was lucky that when I got home from San Francisco, I got back to the Faculty of Medicine at University of Ljubljana. I got a position here, and I was lucky to be awarded a project together with UCSF, Metka Lenassi lab, a big project on EVs. So what we did was we started this EV work and then noticed that there are actually 2 other labs very early on working on EVs. So this is how we organized ourselves and actually started a network of Slovenian researchers on EVs and have yearly meetings from then on. First, it was more in a small scale, like 30 people, and now we actually have 70, 80 people joining us each year to this one-day meeting. So it's quite a popular topic, and hopefully we have contributed something with just education, with connecting everyone, with speaking about EVs locally. So
wow, that's quite a story. I just did a little back of the napkin calculation. That's one EV researcher for every 28,000 people.
This small core group where, because these three labs are really core EV researchers, so mostly working on EVs in different aspects. And obviously we're doing good work of promoting EVs, so many others are involved in EV research mostly. They're interested in a disease and they would like to see it from the EV aspect or use it as biomarker or things like that. So there's different levels of involvement with EVs, but it's funny. We actually included EVs into the main curriculum of the students of Faculty of Medicine. So they actually hear about EVs in the first year, and they also see our lab and stuff like that. So I think it's at least they're aware of it, and I think that's important.
That's really cool. So is there like a week, like a lecture or something spent on EVs for undergrads?
Yeah, so we have there is this one. We had this one module where it's called it's about more science, so medical science, and then they learn about statistics and biomarkers and all of that. And so EVs are inserted there as one of the possible biomarkers, and we have all the students are put into small groups, so it's about 200 or 240 students who then in smaller groups would first hear, let's say, half an hour about EVs and then go to the lab and spend another half an hour, 20 minutes in the lab looking at different ways how you can analyze EVs, just having this, and to at least make them, usually they're very enthusiastic about it, because if you just show them these big cells, these small EVs, and how, what they can all do, I think they're usually quite impressed. It's something, that interests them.
I doubt there are probably very many universities around the world that, do have EVs in their curriculum. That's very cool. Yeah. So I guess, you mentioned as well you were working on HIV. What was the HIV research community like at that time, and sort of where is that sort of what you're still working on?
So they, I think that at the beginning there was a lot of labs that were working on viruses, then extended their research also to EVs. I mean, maybe I exaggerated with the word a lot, but at least some. This was because if you look at HIV, it has in a way many similarities to EVs. So. You could use methods that you used for virus research for EV research. Of course, there was then another level of problem that you would have to separate those two entities, but in the end, I think that it was much easier to get into the field. And even at that time, there were few, even at these annual meetings, there was a section a little bit focused, at least few talks or posters on EVs, but I wouldn't say overwhelming. It's mostly they have, HIV field is just so complex. I mean, they have every level of entry of the virus, every level of, it's just like, it's very huge field and many different interests. But I think that Clotilde Terry also worked a little bit on NEF protein and on HIV, and then there's actually two other people from Matija Peterlin's lab at UCSF who, when they went back to Germany, they also worked a little bit on this topic. So and of course there's many others now.
Is that something you're still working on as well, or where's your current research?
Yeah, so we actually continued with that. So we moved a little bit inside this field into another direction. So our idea was, so mostly in the developed world, mostly people have access to treatment. HIV-infected people are treated mostly. So mostly they have, they're aviremic, so they don't have any viruses in the blood. But still, you see this aging is speed up. So it's early aging, and they have many different problems, medical problems that are just probably caused with some kind of this inflammation processes. And of course, different things can induce this inflammation, or at least expand it or increase it. And what we're interested in is how NEF protein that is transcribed from reservoirs of HIV how it can affect release of EVs and how these EVs that then contain this NEF, how they could promote this inflammation or even neurodegeneration. Because it was shown that HIV reservoirs, I think that probably most are familiar with that. So this is also the reason why this disease needs continuous treatment in most patients, because if you don't treat people with antiretroviral therapy, the virus would wake up. But these reservoirs are actually mostly not working. I mean, they're not producing viruses, more than 90%, because they have some mutations. They cannot transcribe all of the necessary proteins or RNA. So, but what happens is that even though they're not functioning totally like producing viruses, they still, so two important studies show that they can still transcribe different transcripts and also proteins. And they show that NEF is also actually it's transcribed and then translated. So it's there even though the whole virus is not there. And what we've then showed separately is that in half of the treated patients who are aviremic, we could detect NEF in their plasma, although the virus was not present. So we're trying to figure out how this NEF that is part of EVs, how it can affect disease, mostly in nervous system in these patients.
That's really interesting. So is that possibly going to be looked at in a diagnostic capacity?
Yeah, we're trying to, so right now what we're doing is trying to first understand a little bit about, these processes, how, and also, of course, we have hypotheses, we have data that supports some of it, but we want to have enough data to really show that NEF vesicles could actually be important in these processes. But then the end goal is always to then implement this and try to use this in the clinics. So what another part of our lab is doing, so because we are the medical faculty, there was a lot of interest to connect with clinical partners and just use the knowledge that we have on EVs to use it to study EVs as biomarkers of different diseases. And especially two groups that are, so we're collaborating with Dr. Arnold, who is heading the transplantation center for kidneys in Slovenia. And this is one big project we're working on, trying to understand how we could use these EVs that are released from maybe a bit dysfunctional or injured kidney, transplanted kidney, how we could use these EVs in urine to follow up any changes in the transplanted kidney or any changes, is there rejection processes happening or not, before actually doing the biopsy. So to kind of like trying to understand if we can use EVs for continuous measurement in an uninvasive way of what's happening in the kidney. Because the gold standard is biopsy, which is of course invasive. It has many benefits, but you cannot just do it, every month. This is usually after 1 year, unless there are some major problems. So we're trying to fill this gap in between and just use urine, which is very easily accessible, and EVs in there.
I see you've been involved with the urine task force.
Yeah. And maybe what was new in our approach was that we actually were measuring DNA that is bound on the EVs in urine, and using that DNA to look at what's happening in the kidney. Because what's a nice thing there is that, of course, kidney DNA or genome is different from the recipient genome. So we could actually look at specific SNPs that were specific for the donor and compare that to recipient, and we could quantify DNA that came from the kidney and not from anywhere else. So yeah, that was quite a nice approach.
So let's move on to talk about your time when you were previously on the ISEV board as the executive chair of Science and Journals. Maybe just to start off with, what is the function of the executive chair of Science and Journals?
Wow. Yeah, so So this executive chair of science and journals, it's before there was this one big committee of science and meetings that had, was responsible or overseeing all of the meetings that ISEV planned, and then also had this scientific part, which at that point was mostly rigor and standardization activities, and and of course this ISEV guidelines and things like that. So trying to have activities that can improve reproducibility in EV research. But then all of this, so this is where I actually started. So this was probably like a little bit more than 5 years ago. And, it was an amazing thing. I mean, just being involved in all of the meetings, it just gives you a completely different insight. It's stressful, so I'm now really enjoying all the meetings because I don't feel responsibility for everything going very well. But you always had amazing teams of researchers who were really dedicated to make this event the best event ever. So there was a lot of joy in organizing that, and also usually a lot of scientific outcomes. So this was one thing that was part of that. And then the science part, again, is just such an important thing. So this now rigor and standardization subcommittee actually renamed to the Scientific Reproducibility Committee, just so that it's very, very obvious that this is what the goal is, just to improve reproducibility in EV research, and you can do that by many different things and many different actions. And so these activities, this is run by Rink Newland, and I'm sure everybody knows him, so he's very dedicated to that activities. And then there's the whole structure. So many new task forces were implemented in the last few years. So that was, of course, nice to give voice to new let's say new focuses or new topics in the EV field that emerged and needed this support from the community. So I think that we've established a plant EV task force. There's so much happening in plant EVs, also from the therapeutic point. And then there's the, so one is about databases, how to improve or integrate, or what to do with all of these databases, how to use them better, how to make them high quality, how to do that. So this is something recent. And of course also RNA. There's a lot of questions about RNA and EVs, inside, outside, what's happening there. So they will also try to help the community. So this was then rigor and standardization. And we've then established another activity that I think will also have huge impact on the community, and this was special interest groups. So SIGs, we call them SIG. And it's very, the definition is actually very open. It's just group of, not even scientists, I would just say group of people who are interested in specific topic. Let's say we have a special interest group on nervous system, special interest group on genitourinary system, and now the new group is forming, so it will be on EVC in immunity. So there's this, different topics that are then being, where it just helps people to interact and to focus their activities. So the SIG for nervous system and for the genitourinary system, they're probably running now for 2-3 years, and they're very well organized. So we already have, they're already doing these internal structures. They have this, let's say, subtopics that people want to focus on. And one thing is that they will try to write up papers or perspective papers or reviews, or just their feedback on what's happening in the field, where are these key things that we need to address, but then also organizing meetings that are specific on this topic. So there was a huge success with a meeting on EVC nervous system. It was about a little bit more than 200 people. We met in Rome, and it was just, everybody was working on the same topic. Everybody was just so happy to be there because you could really discuss your data inside your group, but still big enough, still very there's so many stuff that were discussed, and people were very open and sharing, and it was just really this great community. So we have so ISEV is organizing another such meeting now in Athens I think it's in November this year, and it will be on EVC immunity, and I've already heard so many people interested, so this will be another smaller meeting, but so 200-300 people, but really focused on a topic, and I think it's going to be an amazing success. And actually the nervous system one is already eager to repeat their success, so they also are organizing one this year in Montreal to just meet again and continue discussions. So these were the special interest groups. So now you see that there was a lot of work already as a meetings and science executive chair that was probably, you can do that for 2 years, but then it just becomes too big. So this is why they separated the science and connected it with journals, which of course, again, makes so much sense because it's journals are also promoting, quality science, promoting they're a vehicle to promote good things in EV research and good studies. And so this was a great fit,
What's the kind of workload like? I guess how many meetings and whatnot are you being involved in? How much are you communicating?
I don't want to put off people from joining the board because actually, it was an amazing experience. And I think it was mostly probably just this committee was really, really intense. But yes, I think that for these 2 years, there is a lot of involvement. But at least to me, it was just so rewarding that for a certain time, this was just such an amazing experience that, you take this into account. But of course, it affects a little bit your time, how that you can spend on research. But again, it was just, I would recommend it if you have, I think that in science, of course, you have people who have, so strong focus on their research and doing excellent work and providing data and studies, but there's also people who are also doing trying to do the best research, but who also feel that they like to maybe are more involved in this community things. I mean, probably it's the same for you. I mean, you're part of SNAP, you're doing this extra stuff, but I feel that there's so many people in the EV field especially who are eager to contribute also in other ways. So I'm sure that this, there will always be a lot of volunteers, and at least I don't know if I mentioned, I'm from Slovenia, from a small country. I mean, there's, like I said, there's quite a few EV scientists per how many people there are here, but
28,000.
But in a way, I never felt like, oh, should I apply or not apply? Is this even a possibility for me? But I can just say that it's really very open. They accept you with very open hands, and they really support you to grow. So I grew so much from when I started as members at large. And then they give you opportunity. I mean, at least this was how it was for me, that a great opportunity came up. I could be leading this activity, and it was just, amazing learning experience.
I can see why you'd be so proud of it. I mean, helping to run these ISEV meetings, and also just seeing how much ISEV's grown and changed with all these new committees and working groups in that short 5 years. It's it's really something. But you
it's also fun, so it's not just work; it's also fun. So we also we made time to also just have fun too.
So you mentioned not questioning whether you should apply at all, but what is the application process like? What's selection and what did that look like for you? Your path through ISEV?
Yeah, so there was usually there's a call out. I think this is probably done. So every 2 years there's reapplication to the board. So, and there's, you can only be part of the board as member at large for 2 times, so for 4 years. And then you have to move to the executive chair position, or, maybe, of course, you can always contribute in different ways, but not as member at large. So there's always some people who leave the board, and there's always openings for new application. So this is just the beginning of the year when there is this new board elected, there would be a call out to all ISEV members to apply. And it's mostly just standard, stuff like, okay, what, of course, also what's your background in EV research, but then also how did you contribute to ISEV activities or to EV research? Did you volunteer to be part of different task forces, or maybe to help write some, perspective on these papers, position papers or things like that? So, or maybe organized meetings. So how did you try to interact with ISEV? And then it's mostly, where do you see yourself contributing and what are your leadership experiences? But it's very open to all of different career levels. We have, I think now there's even, there has to be, okay, hopefully I remember this correctly, but I think there need to be at least 2 representatives of the, more like early career researchers. So, we have, postdocs or, even not people with own established groups. And there's a lot of people are in very different stages. You have people who are already so experienced and then they're mentoring others who come in. And so there's always, even if you're, at the beginning of your career, but you feel that this is something that you want to do, I think this is, it's a good opportunity to interact. And of course, also through SNEV. I mean, SNEV is already, it's a good way of, seeing how these things work and how to get some experience in the activities that you can do. And you're doing so many activities, very well. And you're also, of course, part of ISEV now. So I think this is also one way for young people to get involved and also to, let's say, maybe directly with ISEV.
Absolutely. If you're listening to this podcast, you're probably already somewhat aware of SNEV and probably one of our members. But, if you're not, that's probably a good way to keep in the loop and join a community and consider joining our committee as well at some point if you'd like to maybe jump on this ladder of connecting with other society members and working your way in. So
I can just recommend that to everyone, because really it's SNEV always brings a lot of new energy, new ideas, a lot of volunteers. I mean, I know that everybody is already so busy with their postdocs and worries about the future and stuff like that, but always the joy always comes together with SNEV, so a lot of energy there.
So let's move on to the final questions, which sort of all revolve around this recent Corona workshop. And I was really excited to talk about this because, I think it's, I guess this realization that the field had in maybe, I think it was 2021, that, and this is something that had sort of been speculated upon previously, that maybe there was stuff stuck to the outside of EVs, but, we realized that there is this, sort of cloud shell protein corona around EVs, so that they're not just a little package carrying what was in the cell. They can pick up things along the way. That's quite a profound realization about EV biology. And I mean that's sort of where my knowledge of it is at currently. And I'm really interested to hear what's been happening at this workshop and what's been discovered and where consensus lies. So maybe let's just start off with what the sort of the format of the workshop was, and so who was attending.
So these ISEV workshops, I think, are one of the really, really cool things, really cool events, because the idea is, okay, let's have a very small meeting. This is maybe 50 to 70 attendees, but all are experts in the specific topic. Again, it can be, this can be experts who are, PhDs later in their PhD or early postdocs, but also those who are really established. So it's through all different points of view. And so they come together to discuss a certain topic. And then the structure of workshop is such that you have some introductory talks, but those are mostly meant to just to see, maybe share some ideas, not exactly like, oh, this is my exact research, this is the study that I did, the last study, but more what's your insight into the topic that's discussed through your work. What have you learned through your work? Where are the challenges? Where are the needs? What should we do next? So it's mostly just a start, and then most of the time is spent in these even smaller groups. So maximum 20 people or sometimes even less, who would then openly discuss different challenges inside this topic. So this would then be 2 hours of discussions on very specific problems. Everybody would share. So the format is really, really not just two people speaking up, but just get everyone involved. And that's why it's also very we try to, the format is such that it tries to just, even through the first day, just make it more relaxed, and for everyone, even those who are maybe more reserved, to just bring them into the group and share their thoughts. So it was, and this was the same for the protein corona, or I would say biomolecular corona workshop. And Annalisa Radgeri and Edith Buzas were the co-chairs of this, and I was happy to participate on the IOC, and they did an amazing job, because what they did was they connected experts from the nanoparticle world, so from the synthetic particles, and their experience on on biomolecular corona, which is huge, and then connected it to what people are already doing for the EVs. So the idea was, why should we do the same mistakes and the same things and just go through this several year process? Why not just bring all of these people together to discuss how we could do it better, or at least more focused, or where are the issues, how can we deal with that? Because it's, in a way, it's quite early on. So there's some, there will be a report published, hopefully quite soon, maybe later this year, to report on what's happening. But it was even a success. I think that we had a 1 hour discussion of the definition of protein corona of biomolecular corona for EVs, because it's actually quite difficult. It's much easier for the nanoparticle world. They have this synthetic particle, and then everything that's bound to it, like proteins and other stuff, is corona. Much more difficult for us, there's already, there's plasma membrane with transmembrane proteins. So, are those part of corona or not? It's a bit tricky, but I think that we figured that out, and the results will be shared quite soon.
Yeah. So you guys found consensus on that subject. In general, was there an easy enough time finding consensus on a lot of the points discussed, or is there still a few ones that are ambiguous?
As I said, we were talking for 1 hour about definition.
Okay. Yep.
And this was actually in two groups, so two separate groups, so that everybody could have a voice. And in the end, I think that one group was maybe a little bit more, we have to come out of this really having a definition. Let's start with something, and if we have to improve it later on, we can do it. So let's start with what's already definition of EVs, and then just build on that. So in the end, yes, there was a consensus in the end. So we did reach a consensus, but for many other things, it's it's great that we connected, because we could share data, but there was also a realization that a lot of stuff needs to be done to evaluate. There's no study to really show how and if protein corona affects uptake into, let's say, certain cells or therapeutic activities. So this is, there are some preliminary studies that were shared, but probably I'm not sure that they were published yet. So but this is very early on, so there's a lot of these very core key questions that need to be answered. The only thing that is very definitely true is that EVs do have biomolecular corona, and it changes in different biological fluids, so it could change through time. It's not fixed.
And because you brought up how nanoparticle experts were brought in, is there much that we know about how functionally different nanoparticles are to EVs, or do they just seem to scoop them in terms of picking up a biomolecular corona, or do they just scoop up all of the same things just as easily?
Okay, this is probably I shouldn't say anything because I'm not an expert in this, but let's say I will just, maybe I can comment on my impression of, when I was listening to others. So my impression is that there exists some regulated process of binding of different, let's say, proteins or other molecules on the surface of EVs already through the biogenesis. So this is inside. But then, of course, probably when these vesicles are released into the biofluid, these are just, these different biophysical interactions. I mean, if you have charge, opposite charge, of course, this can attract and can then bind to the surface. So probably there's also some, you could say, unspecific binding in a way that it's not regulated. It's more, there's a lot of this protein that has, exactly the other charge and it binds to the surface. So I think that this biophysical aspect of EVs will be extremely important to understand protein corona, because it's mostly just the strongest bonds, or the dynamics of forming and releasing these bonds is probably very important. But at least some data show that it's also regulated and inside the cells.
Okay, that's really interesting. I know there's this position paper coming out, or this, but I guess the results of the workshop coming out, and you probably don't want to talk about anything too specifically, but I guess could you share what some of the major topics were that were being talked about?
So, of course, was just this biomolecular corona, the definition. How is it formed? Is it formed inside? Is it part of regulated processes? Is this unspecific process? How? So this is in a way how it is formed and from which biomolecules it's formed. It consists. But then there was also part that was interested in, okay, if it's there, how does it affect half-time of these EVs, let's say, inside biofluids? Since if you would use them as therapeutics EVs, how would this affect their half-time? Or how would this affect targeting? So in a way, it was also connected to the function. And then interesting part was also how to use this, or can we use it as a biomarker? Could this, because why not? Of course, if it's there, if it's always there, I mean, it could be an amazing biomarker. Yeah. And one important topic was, of course, also how can we study it? How can we, what are the best methods, or do we need new methods? Do we need new techniques? And again, this in vitro and in vivo, because one comment from the nanoparticle world was, if you do in vitro assays, of course, you will put it in this plasma, or, that urine, or different composition of body fluids. But, what happens in vivo? Because in vivo, there was this one thing that was, I mean, it's obvious, but I was amazed by how that in blood, this is of course not a static current of blood, but it, at least in that model system, it kind of pulsates. So EVs, you should also incorporate that kind of movement into understanding how this protein corona is formed, because it affects this biophysical function of EV. So there's probably in vivo, there's it's just so different. So there was some data presented from labs that were trying to study protein corona in vivo, so that you can really understand what's happening there and in a model system, and not in vitro in the lab, which definitely affects the studies.
Wow, really interesting. I can't wait for this paper to come out. I think that's about all we've got time for. At least that's all the questions I've got for you. Thank you so much, Metka, for agreeing again to speak to us. This has all been really interesting. And is there anything else you'd like to add?
Just saying that it was a real pleasure, and maybe just stressing that I think the EV field is a special field because it's so more connected. I really think that the community is very sharing, very open, and just to motivate everyone to see what their interests are and just try to be more active in this community. I think there's so many opportunities on different levels. So
couldn't agree more. Those are great parting words. Thank you. All right, everyone, that is this episode. Please follow us on all our social medias, and can't wait to hear from you guys again. Bye.
Bye-bye.